Welcome, I'm glad you're here to join in the Mad Forensic Scientist as we go over the basics of toxicology, Fundamentals 101. So what is toxicology? It's a scientific study of the adverse effects of chemical, physical, and biological agents on living organisms and the ecosystem itself. It encompasses the study of toxic substances, including properties, effects on living systems, mechanisms of toxicity, and the ways to prevent or treat toxicological effects. It's a critical field of study that has profound impact on human health and the environment. It plays a crucial role in ensuring the safety of drugs, food additives, and other chemicals. It informs the regulation and management of hazardous substances such as industrial chemicals, pesticides, pollutants, in order to protect human health and the environment. It's an essential field to the fields of forensic science, occupational health, public health, among many others. 5 distinct branches of toxicology include forensic toxicology, clinical toxicology, environmental toxicology, veterinary toxicology, and aquatic toxicology. Now, these are 5 important branches of toxicology, but they're not all-encompassing, as well as there's many divisions within these branches. Now I'm going to dig into some of the types of toxicants. There are chemical toxicants, which are substances that can be harmful and has effects on living organisms through chemical interactions with biological molecules, including heavy metals, pesticides, pharmaceuticals, industrial chemicals, and environmental pollutants. There are also physical toxicants. These are substances that can cause harmful effects on living organisms through physical interaction with biological systems. This includes radiation, temperature extremes, noise, and mechanical injuries. And then there's biological toxicants, substances that can cause harmful effects on living organisms through biological interactions, such as the production of toxins by microorganisms or the venom of animals, such as snake bites and fungi. Bacterial toxins as well, and toxic plants. Common routes of exposure. Inhalation. This occurs when a toxin is breathed through the nose, mouth, and into the lungs. This includes exposure to air pollutants such as smoke or dust, but also smoking and snorting. There's ingestion, which occurs when the toxin is consumed orally. It includes contaminated food or water, swallowing pills, and food or drink spiked with drugs or alcohol. Then there's skin contact. It occurs when a toxin encounters the skin. It includes exposure to chemicals through skin contact in the workplace, handling of contaminated materials, and drugs in the form of transdermal patches or lotions. And then there's injection. It occurs when a toxin is introduced in the body through a needle or other puncture method. It includes accidental needle sticks, drug use through intravenous injection, and occupational exposure to certain chemicals. The dose-response relationship. It's the relationship between the amount of the drug or dose of the toxicant and the resulting biological response. It's fundamental to understanding the toxicity of a substance. It's impacted by several factors, including route of exposure, the duration of the exposure, the timing of the exposure, and the susceptibility of the individual. Other factors include genetic variations, age, and underlying health conditions can also affect this relationship. Understanding these factors is crucial to accurately assessing the level of risk associated with exposure. Linear dose-response relationship increases linearly with increasing doses of the toxicant. Shown here, we have the effect increasing as the dose increases. Then there's nonlinear response, where the response increases disproportionately to the dose. A threshold effect, or a minimum dose required to produce a response. As you see, as the dose increases, the effect does not increase right away, but then once a certain dose is reached, then there's an effect. As well as saturation. So when a maximum response is reached, even with increasing dose. So shown here, even as once a limit has reached of the dose, even with increasing the dose, the effect does not increase. And then there's U-shaped response, which shows an initial decrease with increasing dose followed by decreasing at higher doses. Or it can have the opposite effect, where low dose, there's a high effect, and then a decrease. And then ADME, absorption, distribution, metabolism, and excretion. This is an important aspect of toxicology. Absorption is the process by which a toxin enters the body through one of the routes of exposures. And the absorption rate is influenced by the physical properties of the toxicant, the duration of exposure, and the absorption site. Once absorbed, the toxicant can move through the body to different organs and tissues. And that's distribution. It's the movement of the toxicant throughout the body. It depends on physiological properties, physiochemical properties of the toxicant, the route of the exposure, and biological barriers present in the body. The distribution of a toxicant can influence the severity of its effects on different organs and tissues. So the ability of the drug or toxin to disperse throughout the body is going to have an impact on the effect. And metabolism, the process by which the body breaks down a toxicant into less harmful substances, making it easier to excrete. The liver is the primary site of metabolism for most toxicants, but other organs and tissues can also play a role. The rate of metabolism depends on genetics, age, and health status. And finally, E for excretion, the process by which the body removes the toxicant. Excretion occurs through various routes, including urine, feces, sweat, and even breath. The rate of excretion can vary depending on the physiochemical properties of the toxicant, as well as the health status of the individual. So the healthier a person is able to remove the toxicants better. If the chemical is more lipophilic, then it's more likely to stick into the fat tissues and be harder to excrete. And then finally, target organ toxicity. Target organ toxicity refers to the harmful effects a toxin has on specific organs or tissues of the body. The type of target organ toxicity can vary depending on the toxicant, the dose, and the duration of exposure. Examples of target organ toxicity include neurotoxicity, hepatotoxicity, nephrotoxicity, and cardiotoxicity. The severity and type of toxicity can be influenced by the toxicant's physiochemical properties, the dose, the duration of exposure, and the individual's health status. So much like Paracelsus said, the dose makes the poison. As you can see here, the dose varies everything. Other factors include the route of exposure, the age, the sex of the individual, as well as the presence of other chemicals. The specific types, neurotoxicity, refers to the harmful effects of the nervous system, including the brain, peripheral nerves, and sensory organs. Hepatotoxicity refers to the effects on the liver, which includes liver damage, inflammation, as well as impaired liver function. Nephrotoxicity refers to the harmful effects of toxins to the kidneys, which includes kidney damage, inflammation, and impaired kidney function. Cardiotoxicity refers to the harmful effects of the toxin on the heart, which includes damage to the heart muscle, impaired heart function, and irregular heart rhythms. Some mechanisms of toxicity. So there's direct toxicity, which refers to harmful effects the toxin has through direct interaction with cellular components, such as DNA, proteins, or membranes. It can lead to cell death, tissue damage, organ dysfunction, and examples of this include heavy metals, pesticides, and solvents. There's also indirect toxicity, which are the effects that are caused through an indirect mechanism, such as reactive metabolites, disruption of cellular signal pathways, and modulation of immune function. This can result in a wide range of adverse health effects, including cancer, immunotoxicity, reproductive toxicity, and there's examples of this found in drugs, environmental pollutants, and other toxins. So some drugs have metabolites that are active. So dextromethorphan actually has a metabolite that is. So dextromethorphan, if you don't know, it's cough syrup. It helps produce as a cough suppressant. So it's given normally in cough syrup. However, in high dosage, dextromethorphan actually has a metabolite that is a psychogenic. So it actually will cause hallucinations. So that's an example of a drug that has a reactive metabolite. And there's receptor-mediated toxicity, which refers to the harmful effects of toxin that occurs through interaction with specific cell receptors. So this toxicant can bind to receptors, which activate or inhibit signaling pathways, which leads to a wide range of health. effects. Which we've gone into the final aspect of the basics of toxicology, which is extraction techniques. Extraction techniques are an essential component of toxicology analysis, which allow for isolation and purification of analytes from complex matrices. A toxicologist is going to be focused in on getting the drugs or the toxins from the biological components so that they can analyze them and identify them and quantitate them. One key extraction is solid phase extraction, or SPE. It's an extraction technique that utilizes a solid phase adsorbent to extract the analytes from a liquid sample. The analytes are absorbed into the adsorbent and then eluted to with a suitable solvent. SPE is a preferred in toxicology analysis due to its high selectivity, sensitivity, and reproducibility. So shown here is a SPE cartridge. You put your liquid sample into the SPE tube, rinse through it with solvent. The drugs that you were trying to extract are adhere to the solvent, and then we use a solvent to push it out. So a solvent that the drug has an affinity to withdraw the analyte. I'm actually going to do a presentation later to go into more in depth for the extraction techniques, but this is a basic introduction. Then there's liquid-liquid extraction, or LLE. This is a classical extraction technique that separates analytes based on differential solubility into two immiscible solvents. So in liquid-liquid, the analytes are partitioned between an aqueous and an organic phase and are often recovered in the organic layer. LLE is useful in toxicology and extraction of nonpolar analytes from complex matrices. So you have an organic phase and an aqueous phase. Usually it's a harsh organic, like MTMB or hexane. They will not partition to the liquid. So as you realize, I don't know if you realize, most of your body is water. So if you choose to use a harsh organic solvent that is not miscible with water, then you will have an extraction. So you can use the characteristics of the drug to partition the drug into the organic phase. Then you have protein precipitation, which is a sample technique that removes interfering proteins by precipitating them out. Then they recover from the supernate, typically by centrifugation or filtration. It's a commonly used extract from small molecules. This is what we often call dilute and shoot. So we just put a solvent into a small amount of sample, usually urine or saliva, and we crash out everything, crash out all the proteins and cells, and leave behind the drug in the organic. And then we take off the organic and we run that on the instrument. And then we have matrix solid phase dispersion, or MSPD. This is actually basically a catcher's extraction. So it's a novel technique that combines the principles of solid phase as well as mechanical disruption. Extraction techniques are very important for toxicology analysis. It's important to choose the type of extraction depending on the type of analyte you have, the type of matrix you have, and the selectivity of your detection method. It's very important. This is the kind of things that a toxicologist has to think about. The physiochemical components of the analytes that we're trying to detect, the matrices that we're trying to extract the drugs from, as well as the instrumentation that we are using to detect. All that impacts our choices for toxicology analysis and our extraction techniques. So it's all very important. This was all just an introduction to toxicology. We're going to go into more detail of the chemical analysis and what we use to decide these extraction techniques in future episodes. So I hope you guys enjoyed that, and if you have any questions, now is the time. Once again, thanks for watching. Hope you enjoyed it. The Q&A session will be posted in a future time. If you like what you saw, be sure to hit that subscribe button to follow for more shorts content, as well as more info from toxicology from the Mad Forensic Scientist. Appreciate you guys.